Hello students, I'm Sonia Nan, your biology educator for class 10, and today we are starting a new chapter, that is heredity, and today we will be covering part 1 of heredity chapter, that is heredity and Mendel's laws of inheritance. Welcome to the class, hope you all are doing well. Now let's start the chapter. First, here we have the content of chapter. What we are going to cover today: heredity. We will see what is heredity, then variations, important terms, and key concept to understand this chapter in a better way. Crosses, monohybrid cross and dihybrid cross, Mendel's work, laws of inheritance. Before I start this chapter, let me tell you an important thing. You might find this chapter very hard. Okay, if you just try to mug up this chapter, sorry to tell you guys that it is impossible to mug up this chapter. If you want to cover this chapter, you have to understand the basics of the chapter, the important terms we are going to discuss in this chapter, and then you have to understand each and every topic of this chapter. Only then you can remember this chapter. There is no any other way to remember this chapter except grasping the basic terms and understanding each and every concept of the topic. We'll be discussing first important terms and then we will discuss topics. Okay, don't try to mug up this chapter. Again, I'm telling you this thing. Don't worry, I'll try to make these terms and concepts very simple for you so that you can remember these things in an easy manner. Okay, here first we have heredity. What is heredity? As we are studying chapter heredity, we should be knowing that what is heredity. Heredity is the transfer of characters or traits from the parents to their offspring. This process is called heredity. Heredity is transfer of characters. Now what are these characters? What are these traits? We have to understand this thing. We know parents give rise to their offspring. Offspring means their child, or there is another term for offspring in genetics. Here we have this progeny. You have to remember these terms. What is offspring? What is progeny? Okay, offspring is child. We use this term offspring for child in genetics. Okay, in biology, and this term is also used progeny. Okay, offspring, progeny. Parents give rise to their offsprings and progeny. This much is clear. All right. Now, parents give rise to offspring and progeny. When this process happens, there is transfer of characters, transfer of certain characters or traits from parents to offspring. Okay, certain characters, characteristics. All right. Okay. What are characters? What are traits? Characters or traits are the things which are visible to us. Okay, like our skin color, our hair color, all these things are characters, our height, all these things are character, and these character are transferred from parents to their offspring. Okay, I'm writing down here example of character like height. All right, height. So the character of height is transferred from parents to their offspring progeny. Okay, if the parents are tall, chances are high that their child and offspring will also be tall. All right, understood the logic behind character and heredity. Heredity, you have to just write that heredity is the process of transfer of characters from parents to their offsprings. This is heredity. Very simple. Understood? Okay. Transmission of genetic information occur with the help of this process, heredity. So heredity is important for transmission of genetic information. Why heredity is important? For the transmission of genetic information. One line answer. All right. What is genetic information? What is genetic information? Genetic information is the information which is present in the genes, which is present in the genes. Okay, genes are present in our DNA. Okay, so we can say that it is the transmission of genetic information in the form of DNA from parents to their offspring. This process is known as heredity. Now, genetic information is transmitted through gametes, sperm, and egg cells during sexual reproduction. This you have read in your reproduction chapter, that how this thing happen. Genetic information is how now we know the definition of heredity. What is heredity? Heredity is the transfer of genetic information from their parents to offspring. Now we have a question here that how this information is transmitted from their parents to offspring. How this thing happen? Okay, how a parent transfer its genetic information, its genes to their child, to their offsprings, progeny. How this happen? So this thing happen during the reproduction. So genetic information is transferred through gametes like sperm and egg cells. These are the gametes. Sperms are produced by males and egg cells are produced by female. Okay, and during fertilization these sperm and egg will form a zygote, and then that zygote will give rise to a newborn baby. Okay, this is the thing. And during this process, during this gamete formation, genes of parents, gene of father and gene of mother are transferred from parents to their offspring, to their progeny. So this is how the transmission of genetic information occur from parents to their offspring. One line answer is through sexual reproduction. Okay, through sexual reproduction, during sexual reproduction, and this information is transmitted through gametes. Okay, understood? Through gametes, sperm and egg cells. Okay, now you know what is heredity, and now you know how the transmission of genetic information occur. It was very simple, right? Now next we have this another important term. Next we have this another important term, variations. What is variation? Variation in very simple term is the differences. We can write here variations are the differences. Now we have to remember this word differences, and everyone knows what we understand from this term differences. Okay, this is a very common term. Now, variations are the differences between the characters or traits among the individuals of the same species are called variations. Now, species, this is here we have one term species. Everyone knows what is species, like human. We human are a type of species, dog are another type of species, cats are another type of species. So we have so many species. Let's consider for now species of human. If you see here, there is difference between the characters or traits among the individual of the same species. Okay, we are a species, human. Now all humans are not same. There is difference in skin color, hair color, height. There are so many differences in different human populations. Okay, people living here in India and people are from other countries like from US, from South Africa, from other parts. We have differences. We have so many differences in our body structure, in our appearance. Okay, and those differences which are present in between human, in between same species, in between individuals of same species, are considered as variation. If you can correlate this thing with the picture given here, you can see that their skin color, if you notice their skin color is different. Now, this difference in skin color is variation in skin color. So skin color is a character, is a trait, and there is difference in this character in the population of human, and human is a species. This difference is variation. The differences between the characters or trait among the individuals of the same species, these are the individuals of same species, but they are not same. They have minor differences, like they have difference in their skin tone. So this difference is variation. Now you know what is variation. This is one example of variation in human. Example of variation is this skin color in human throughout the world. This is variation. Now, accumulation of variations during reproduction. When organisms reproduce, the offspring show minor variation due to inaccuracies in DNA copying. Now how this thing happened, that how these much variations are present in the populations of the same species? How does this happen? Which process is responsible for causing variations in the population? why these variations are present in the individual of same species, why they are not same, what is the reason behind these variations. If we try to know the reason behind these variations, why there are variations and what are the importance of variations, what we have during when the organisms reproduce, we know this thing that the DNA is copied. We have DNA in our cell, and during the cell division, this DNA is copied. Means a same copy is formed of parent DNA. This is parent DNA, and then during the DNA replication. This is the process of copying DNA. This exact DNA is copied in a new DNA and a new DNA is formed. This is the parent DNA and this is the new DNA formed after copying of this old DNA. So this is DNA replication. So whenever cell division take place, this process happen. This process of DNA copying. So we have here DNA is copied. Now, when this DNA is copied, there are certain inaccuracies in DNA copying. Although the process of DNA copying is very efficient process and many a time it is very accurate, but chances are always there that there are some few inaccuracies, very minor inaccuracies in copying the DNA molecule. If there is any inaccuracy in copying the DNA, okay, that will lead to variation. If there is a very minor inaccuracy in copying DNA, that minor inaccuracy will be transferred to the offspring, and now that inaccuracy will lead to a different character. It will modify that character. Like in previous example, if we consider skin tone in case of human across the world, let's say in the parent cell when the DNA is copied and there are few inaccuracies and that inaccuracy leads to darker skin tone, then over the generations this variation will accumulate. Okay, this variation will accumulate means this variation is transferred from generation to generation, and over the generations this accumulation will lead to a variation in population. DNA is copied and here we have a minor inaccuracy in DNA copying. This inaccuracy is transferred over generation, transferred to their son and next to their son. This is transferred over generations, and if this happen, this will lead to the accumulation of these variations in the population of same species. Now if there is accumulation of variation, this will lead to a difference in this character. So when organisms reproduce, the offspring show variations due to inaccuracies in DNA copying. This is the reason that variations are caused in the population. These minor variations which are due to DNA copying will lead to differences in the character over many generations, okay, which happen during cell division, during reproduction process. Next we have this, now here we have this example of variation. Here in this picture we have this concept of variation, how the variation is created, variations, and this variation will lead to diversity. Diversity means differences. Let's say this is the parent. I write down here P to represent parent. Now this parent will give rise to two offspring, 1 and 2. They are similar to their parent, but there is a minor difference, subtle difference between these two progeny, between these two offspring. They are different from each other and as well they are different from the parent. You can trace the difference here. This is the difference. This is the difference. A difference is created. Now this change is carried over the next generation. As this change is transferred to the next generation, and this change is also transferred to the next generation. Now this is the change which is transferred from the previous generation, which is transferred from this generation. Here we have this change, but here we have few more changes. These changes, this is different from this parent and also from this member. So here we have again new difference and the difference of previous generation, created in previous generation. Same way here we have this change and this change is transferred to these offspring. We have these changes, this change and this change from the previous generation, from this generation, and this change is created in this particular generation. So what I'm trying to say you is These changes are created over the generation, accumulated over the generation, and along with these changes which are transferred from generation to generation, some differences, some changes are unique also. Unique means which are created in that generation only. Like if we are saying that this is the change which is transferred from this generation to this generation. But now here we have another new change. This change is unique to this generation only. Okay, unique to this generation only. And now this difference, this change is transferred, will be transferred to next generation. And this process keep repeating itself over the generation. If this process keep happening, what will be there? Accumulation of variation, and this accumulation of variation will lead to diversity. This is how the variation concept works over the generations. The generations variations are accumulated, and this will give rise to differences in characters. Okay, so this is the concept behind variations. Now you know that this is parent, some differences are there. Okay, then in next generation these differences are transferred, along with there are some unique differences which are unique to this generation. Here we have example of human population. In human population, you might have noticed that we have this ear, lower part of the ear. Let me show you this lower part of the ear. Okay, this is earlobe. What is this? This is earlobe. Earlobe is attached to our head. In some cases, this earlobe is attached to our head, completely attached to our head, like this way, like you can see in this picture. This earlobe is completely attached to head region. But in few cases, this earlobe is not attached to head. This is free. Like this is free. In my case, this is free. You can check yours also, like your earlobe, whether it is free or not. In some cases, this earlobe is attached, while in other it is free, and in some cases it is attached. So here we have difference in earlobe. This difference in earlobe is accumulated over the generation. This gave rise to variation in earlobe attachment. So this is the perfect example of earlobe variation. And this example of variation is given in your NCERT. So this is the example of earlobe attachment in human population. This is about heredity and variation. So here we have the key concepts. Okay, these are the terms which we are going to use in upcoming slide. I'll be using these terms in upcoming slide, and here I'm going to explain you these terms so that we can use these terms in later part without any explanation. What is trait? Trait, a specific characteristic or feature of an organism. Trait is any character or feature of an organism. What like feature? Like body structure, body appearance, or external appearance is trait. Like trees are green in color, leaves of tree are green in color. This is the trait of tree, that leaves are green in color. Our hair are black or blonde, so this is the character of our hair color is character. Okay, height, height of a person is character. So these are features. You know what is trait? Trait is any feature of the organism. If I give you example of trait, like our eye color, our hair color, our height, any other body appearance, that is considered as feature, trait. Now we can write down here the example of trait. We can write down here hair color, like in case of plant, flower color. Genes. Genes are the segments of DNA that carry instructions for specific trait. Gene. What are genes? Genes are DNA. Let's say this is the DNA. DNA is present inside the nucleus of our cells. What is gene? Let's say this is our DNA. And in this DNA, this region has a certain function. Let's say it determines flower color. Let's say this region of DNA determines flower color. This is DNA, a very long chain of DNA, and let's say this region, okay, is responsible for flower color in case of plants. This region is a gene. Okay, this segment is a gene, and this gene is responsible for flower color. So genes are the segment of DNA that carry instruction for specific trait. means this is the region of DNA that have a certain function. In this example, we have considered flower color. So the region of DNA which will give color to the flower, that region will be known as gene of flower color. Now there are many genes present in the DNA of an organism, and those genes are responsible for various functions. Okay, each gene has its own function. Like if we consider in case of plant, flower color gene, what will flower color gene do? It will provide color to the flower. Okay, it will be responsible for color of the flower color gene. Same way we have the height gene. Same we have gene for height. Okay, let's say T, this region. Let's say this region of DNA, this segment of DNA is responsible for height of the plant. So this gene is responsible for height trait, and height is a trait. This segment of DNA is responsible for height, and height is a trait. So we can use gene term for this. So this is the gene responsible for height of the plant. Genes are the segment of DNA. These are segments like this, and they have certain information inside this segment for a trait, for specific trait. Every gene has its own function. They control a particular trait. Like gene of height will control height, gene of flower color will be determining the color of flower. Same way we have so many genes. Okay, these were the examples of gene. You have to explain gene as a segment of DNA, and this segment is responsible for a specific trait. What is trait? Trait is a feature or character of an organism. Okay, that feature character can be anything. Here we have this chromosome. What are chromosomes? Chromosomes are the structure that contain genes and are located in the nucleus of cell. There are few things like we have DNA, okay, gene, chromosomes. These are DNA only. When I say DNA, you understand DNA. And when I say gene, gene is also DNA. But here what is the difference? That this gene is specific part of the DNA which determine specific trait, which is responsible for a specific function. This segment of DNA, this whole DNA, and a short segment of DNA is gene. Gene is also DNA, but it is a short segment of DNA. Now, when I say chromosome, chromosome is also DNA. DNA in our cell, DNA in the cells of an organism is folded, and this folded DNA is known as chromosome. Okay, so chromosome is the folded form of DNA. Chromosomes are the structure, and these chromosomes contain genes, and they are located in the nucleus of cells. Now you understand what is chromosome, and on chromosomes we have genes. Okay, and genes are the segment of DNA. Chromosomes, DNA, and this DNA folds to make chromosome. Now this is our chromosome, and on this chromosome we have certain regions which determine certain function, certain feature. Those regions which are responsible for a trait are known as gene. They are the segment of DNA which are present on chromosomes. Now we have this alleles. Alleles are the different form of a gene, and these are called alleles. Now we know gene. Okay, so here for each gene we have two different forms. We have two different forms for a single gene. Like consider the gene for height. Here we have the gene of height. Okay, in case of plant, let's say this is the gene of height. Okay, plant height. This gene will have two different form, capital T and small t. Okay, will have two different form. You just have to remember that each gene has two different form, and those two different form are known as alleles, and these two forms, capital T and small t, are the alleles. Here we have plant height gene. So alleles are the different form of a gene, and these are called alleles, which can result in variation of trait. Okay, now this allele will determine height of the plant, and this allele will also determine plant height. But what is the difference between these two form of gene? This will lead to tall height, and this small t will lead to short height. This is the difference. Tall and short are the two form of height. If a person is tall, we are discussing height. And now if I say a person is short, Again, we are discussing on height. So these are the two different form of height. One is tall, other is short. So same way we have a gene for height, and that gene can may have two form. First, a form which is responsible for tall height, and the second form which is responsible for short height. Okay, and those two form of a single gene are known as alleles. We have a single gene of height, and this gene of height can have two forms, one responsible for tallness and another responsible for shortness. Understood? So alleles are the different form of a gene, and these are called alleles. Or whenever I use a term, again, whenever I use another term, very important, factor. Factor is also allele. Allele and factor both are the same thing. Whenever I use factor, you have to understand that I'm discussing alleles, okay, or alleles or factor. You have to remember both are same. And normally we have two form of a gene, homozygous and heterozygous. What is this homozygous and heterozygous? As we have studied in previous slide, that we have a gene, plant height gene, okay. This gene is responsible for plant height feature. Plant height, this is trait. This gene is responsible for this trait of plant height. Now this gene can have two different forms, capital T and small t. These are alleles. And for example, here I have taken plant height. What is homozygous and heterozygous? Homozygous is when the organism have two copies of the same allele for a gene. Homozygous condition. Homo means same. In case of homozygous condition, an organism will have same type of allele in their cell. Whenever you find this double T, what it denotes? It denotes that this cell have T, capital T and capital T, both same type of alleles. If a cell contains same type of alleles for a trait, then that will be considered as homozygous. If an organism contains heterozygous, means hetero means different. Organism will have different type of alleles in their cell, will have capital T and small t, different alleles for a trait. These are the alleles of plant height, and this is also the allele of plant height. You know what is homozygous and what is heterozygous. In homozygous, we have two possibilities, capital T, small t. But in case of heterozygous, we have this possibility, capital T, small t. Okay, heterozygous, two different allele of a trait. Clear? Now, the organism which has two different allele of a gene is heterozygous, capital T, small t. So for now, you have to just remember what is homozygous, what is heterozygous. So here next we have dominant trait and recessive trait. Trait is feature like height. Height is a trait. This we know. Now, trait that will express phenotypically in the heterozygote. What is phenotypically? This is again a new term, phenotypically. Phenotypically means external appearance. Means external appearance. External appearance is phenotypically. Phenotype is the external appearance. Like if we say, if we see a plant, if we see height of plant, there are plants which are taller and there are plants which are shorter. This is the appearance of plant. We can see that this plant is tall, this plant is short. So this is the phenotype of plant. This tallness and shortness is the phenotype of plant. This is the external feature of the plant, and the external feature of the plant which we can see is phenotype. Okay, so whenever I use this term phenotypically means the thing which we can see, which we can visualize. For example, flower color is a phenotype, as we can make difference between the color of flower. So flower color is a phenotype. It is the external appearance of a flower. We have trait that will express phenotypically in heterozygote, and a dominant allele is expressed by capital letter T. There are, for example, in case of plant height, we have two type of traits. We have two type of trait. One is tall, okay, tall plant, and second is short plant. These are two traits in case of plant height. Out of these two traits, one trait is dominant and the second trait is recessive. Dominant, recessive. The dominant trait, as the name suggests, it will be dominant. Okay, no matter what the condition is, it will show its expression. This trait is always expressed. And the short, this is recessive. This trait is not expressed in the presence of dominant trait. If there is dominant trait, then that trait will not will overexpress the recessive trait. Here we have two things: dominant, dominant versus recessive. Okay, dominant is strong and recessive is weak. We have two type of alleles: dominant D and recessive R. Dominant always express itself, but recessive expresses only when this is present alone. Let's say condition of plant height. For plant height, we have two different alleles, capital T and small t. Now this capital T is dominant, this capital T is dominant, and this small t is the recessive allele. This dominant allele will be responsible for tallness of the plant, and this recessive allele will be responsible for the short height of plant. Let's say in case of plant 1, we have this we have capital T, and in case of second plant we have this condition capital T small t, and in third case we have this situation small t. Now, as I have said you, the dominant allele will always express itself. So this plant will be tall. And in this case, as capital T and small t are present together, in this case, only capital T will express itself. T will show its effect. Okay, so it will make plant tall. So this plant will also be tall. Now in this case we have both small t. We have both small t, and small t is responsible for short height. So this plant will be shorter in height. So what I'm trying to convey you is when capital T means the dominant allele are present together, then this will lead to dominant character, that is tall height. But if dominant allele and the recessive allele are present together, then dominant allele will express itself and will lead to the dominant trait, dominant character. And in this case, we have dominant character, tall height. So this plant will be tall. And in third case, we have both alleles, small t. Okay, small t means recessive alleles. And these recessive allele will be responsible for recessive trait, which is short height. So this plant will be shorter in height. Understood the logic behind dominance and recessiveness. So if I move to the previous slide, here we have that a trait that will express phenotypically in the heterozygote. In the heterozygote means this situation. The trait which will be expressed in this situation is dominant. And in example, we have seen which trait will be expressed in this situation: tallness. Okay, tall. Plant will be tall. So here the tall is the dominant trait. Understood? Now, a dominant allele is expressed by capital letter T. Whatever trait we consider, always we write capital T for the dominant allele and short letter for the recessive allele. So we use capital letter for the dominant allele, and for recessive allele we use short letter. Understood? Okay. So now recessive trait. Recessive trait is trait that will only express in homozygote. This trait will express in homozygote condition, and in which homozygote condition? In condition where only recessive allele is present. Where only recessive allele is present. In this condition only. Small t. Okay, and we represent recessive trait by small letter. As in case of plant height, we use this small t. What is dominant? What is recessive trait? Next, we have these crosses: monohybrid cross and dihybrid cross. What is cross? Cross is this is cross. This is parent. This is parent. This also parent. Okay. Now, in a monohybrid cross is a genetic cross that involves the study of inheritance for one trait at a time. Crosses are for study of trait, how the trait is transferred. Now, let's consider, for example, we have trait of plant height. We have these letters, capital T and small t, one trait at a time. And in this monohybrid cross, we consider only one trait at a time. Means we will be studying one trait in a cross. So here we have only one trait, that is plant height. Now in this case of plant height, I have considered only this trait, and here we can write down that this is parent 1, this is parent 1, and this is parent 2, and these are the alleles present in the cell. If we cross them, what will we get? How do we perform this cross? We perform this cross as, if I try to write it down like this, we perform this cross such a way, in this way, like we will have this, then we perform this as, okay, we will get this. Now in this case what we have, then this T is crossed with this T, what will we get? We get this, and then this is crossed with this. Okay, so this is what we get. This is monohybrid cross. Okay, in which we have considered only one trait, like we consider plant height trait. These are parent 1 and parent 2. These two parents are crossed, and after crossing we have got these results: T, T, then T, T, again this T, this T, this T, this T. So these are the result. And in dihybrid cross is a genetic cross that involves study of inheritance for two different traits simultaneously. In previous case, we considered only one trait, that is plant height. Okay, but in case of dihybrid cross, we have two traits at a time. Like if we consider two trait, one is plant height and second trait is seed shape. Okay, shape of seed. Now for plant height we have these alleles, and for seed shape we have these alleles. Round, R is for round, and small r is for wrinkled. These are two traits, and these two traits are considered at a in a single cross. This dihybrid cross and monohybrid cross we will discuss in upcoming slide in detail. This is just to make you clear that we have two type of monohybrid. We have two type of crosses: monohybrid cross and dihybrid cross. In monohybrid cross, we consider only one trait at a time, like I have told you with the help of example. But in dihybrid cross we consider two trait at a time. Di means two, hybrid means cross. So with here we have two crosses. Next we have generation. What is generation? Generation refers to a group of individuals with a common set of parent or a common ancestor lineage. Generation is a group of individuals with a common set of parents. With a common set of parent. Let's say this is the parent. Okay, this give rise to these two offspring. Now this is generation 1. Now this we have parental generation, parent generation, and now this parent generation give rise to this generation. This is generation 1. Further on reproduction, will give rise to this will give rise to second generation. Parent generation give rise to first generation, then first generation further on reproduction give rise to second generation. So generation refers to a group of individual with a common set of parents. These two individual have this common parent, these four individual have these common parents. Okay, so generation refer to a group of individual with a common set of parent or a common ancestral lineage. You have just to remember this thing, common set of parents. In case of first generation refers to the offspring produced by crossing two purebred individual from parental generation, P generation. This is P generation, parental generation, and it will give rise to generation 1. Parental generation is pure breed. Pure breed means that it contains same type of alleles in the cell. Either it can be dominant allele or it can be recessive allele, but it contains homozygous condition in the cells. So this is pure breed. In second generation we have, this refers to the offspring produced by crossing individual from F1 generation. Now once we get F1 generation, like this is our F1 generation. Now the individual of F1 generation, these individuals, individual 1 and individual 2, these are the individuals. Okay, they are crossed, and after they are crossed they will reproduce this generation, second generation. This is the second generation. First generation, second generation, this is parental generation. Parental generation will give rise to first generation. This is first generation. Further on crossing these two individuals of first generation, we will get second generation. Here we have this diagrammatic representation of F1 generation and F2 generation. In F1 generation, if you see here, we have a plant which has capital capital T alleles, and in case of short plant we have small t. If we make a cross between this capital T and small t, we will get this after crossing. Okay, this we have studied in previous slide. How do we get this thing? Now this generation capital T small t is parent generation. This is parent generation, or in this diagram they have considered it. So this is parent generation. Okay, and upon cross in parent generation, what will we get? F1 generation. Now this from this F1 generation we again have a cross between these two individuals, and on crossing these two individuals of the F1 generation we will get F2 generation. So we can write down here that F1 into F1 cross will give rise to F2 generation. This is a parental generation, F1 generation and F2 generation. You know what now, what is parental generation, what is F1 generation, what is F2 generation. First parental generation, first generation, second generation. Okay, this is parental generation, this is first generation, and on crossing these first generation offspring we will get F2 generation. Now in case of F2 generation, here we have this situation, capital T small t, capital T small t, and these are the result of second generation. These are the offsprings of second generation. Capital T, small t, capital T, small t. How did we get these? Capital T, small t. We made cross between these F1 generation offspring, and that's how we cross them. Capital T, capital T. Next, this capital T, small t. Okay, let me change the color. Next here we have this. Okay, capital T, small t, then this small t. Okay, this is our F2 generation. Now you know monohybrid cross. Now you can perform monohybrid cross, and you know what is parental generation, what is F1 generation, what is F2 generation. Okay, all right. This was your basic. Okay, now here we have very important topic, Mendel's law of heredity. Now we have discussed the basics of the chapter. Here we have the important topic now, Mendel's laws of heredity. Okay, Gregor Mendel was a scientist, famous scientist and mathematician, and he worked on genetics. He worked on the principles of heredity. Okay, he has proposed this work of heredity and these laws of heredity, what we are going to cover in this chapter. So Mendel is considered as father of genetics. Father of genetics is Mendel. Now, about his work, Mendel has performed certain experiments on genetics in 1856 to 1863, and he conducted the experiments on the garden peas. So here this is the diagram of garden peas. Okay, these are peas, and he performed experiment on these pea plant. Now Mendel used a number of contrasting visible character of garden peas for his experimentation. What are contrasting traits? We know what are traits, right? Trait is any character or feature. This we have understood. Now, what are contrasting traits? Contrasting traits are the traits which are clearly visible to us, that we can differentiate between those characters. Okay, we can clearly differentiate between those two characters. All those characters are contrasting characters. Like if I consider here flower color. In case of peach, we have two different flower color. One is violet, okay, and second is white. Violet and white. Now you can easily differentiate now that this color is violet and this is white. So he considered only the contrasting trait for his study, for his experimentation. For example, height. You can easily tell that which plant is tall and which plant is short. So again, this is a contrasting trait. Now you know what is contrasting trait. Okay, contrasting trait are the traits which are clearly visible to us, so that we can make difference between them. All right, so we have these contrasting traits, and Mendel worked on contrasting trait. For example, if we see There in pea we have two type of seeds, round seeds and wrinkled seeds. Plant height, tall and short plants. Flower color, white and violet, and so on. He considered many characters, many contrasting characters for his experimentation. If we discuss about Mendel's work, Mendel's experiments, Mendel performed two type of crosses, monohybrid cross and dihybrid cross. And this we have discussed in our previous slide, what is monohybrid cross, what is dihybrid cross. So during his experiments, what he had done, he had performed these two type of crosses, monohybrid cross and dihybrid cross. Okay, he considered contrasting trait for these crosses. He performed monohybrid and dihybrid cross for plant height and then analyzed how this plant height trait is transferred from parents to their offspring or progeny, how the plant height character is transferred over the generations in pea plant. After all this experimentation, he concluded certain laws of heredity, and these laws tell us that how the characters, how the traits are transferred from parents to their progeny. Now, what are the laws of inheritance which are concluded by Mendel? First is law of dominance. Second law is law of segregation. After that, third law is law of independent assortment. Law of independent assortment. This is very simple if you try to understand that Mendel performed certain experiment on pea plant. Alright, this we know. And he worked on contrasting visible trait. He took a trait like plant height and then performed monohybrid cross and dihybrid cross for that to study that trait like plant height. And he studied this trait with the help of monohybrid cross and dihybrid cross. Same way, he considered another characters also like flower color and seed shape characters, contrasting visible characters. After all this experimentation, finally he concluded certain laws based on the analysis of experiments. So what he got this law of dominance, law of segregation, and law of independent assortment, and these are important for your examination. So now we are going to understand what is law of dominance, law of segregation, and law of independent assortment. These laws are given by Mendel. Okay, this is Mendel's work. Now, law of dominance is, this states that in a pair of contrasting trait, you have to mention this term contrasting trait. In a pair of contrasting trait, one trait may dominate over the other. Let's say we have a character of this flower color. Now we have two type of color, violet and white. Flower color is trait. Let's take the example of trait, and trait we have here is flower color. In flower color we have a pair of contrasting trait. In pair one we have violet color and second we have white. One trait may dominate over the other. Now this we have trait, this is also trait, but out of these one will dominate over the other. One will expressed in the presence of other. But I'm trying to say that, let's say if we have this violet, okay, we are representing violet by capital P and white by small v. If we have this condition, heterozygote condition, capital V, small v, then this plant will be violet color. Why? Because this V is dominate over small v. Violet color is dominate over white color. So whenever we have allele of violet color, that is capital V, and allele of white color, that is small v, in the same plant, then that plant will be having violet flower color, because V is the dominant allele which dominates over recessive allele small v. White is recessive in this case and violet flower are dominant. So this is the law of dominance, that in every cell, in every organism, for a given trait, two factors are present, two alleles are present. Now out of those two alleles, one is dominant and another is recessive. The dominant trait is expressed in the offspring when an organism carry both dominant and recessive allele, heterozygote. If this is the situation that an organism is carrying both dominant as well as recessive in their cell, now what will happen, this dominant will be expressed in that case. Okay, let's, as I have told you that in a plant if we have both allele for violet color and allele for white color. Then what will be expressed? Violet one will be expressed because violet one is dominant. So this is the law of dominance, that the dominant trait is expressed in the offspring when an organism carry both dominant and recessive alleles or factors. You can write down here factors. Okay, now this is the example, and this diagram you have to draw whenever you explain law of dominance. Here if you see, here we have the parental generation. These are the parents. One is violet and second is white. For violet we can write down here capital V, capital V, and for white color we can write down here small v, small v. On cross fertilization, what will we get? We will get F1 generation, and F1 generation will be capital V and small v. Now, this is capital V and small v. After self fertilization, what will we get? Self fertilization means V, this same is crossed. Okay, this is self fertilization. On self fertilization, we will get, this is capital V and this one is capital V, small v, capital V, small v, and this is small v, small v. This capital V will be tall, and this capital V, small v will also be tall because capital V will dominate over small v. Violet will be dominated over white. So this will be violet, and this also be violet, capital V, small v, and this one is white, as this contains only small v, white allele. Okay, recessive allele. In the absence of dominant allele, recessive allele will express its expression, but in case dominant allele is present along with recessive allele, then only dominant allele will express its expression, like in case of violet and white. So ratio here we get is 3:1 ratio, violet flower and white flower. We have this ratio, 3:1 ratio. Out of 4 offspring, out of 4 plant, we will be having 3 violet flower. We will be having plant which have violet colors and one is white. So this is law of dominance. And you have to write this only, that for a given trait in each organism there are 2 factors, and a pair of contrasting trait or a pair of factor, one factor may dominate over the other or one trait may dominate over the other. The dominant trait is expressed in the offspring when an organism carries both dominant and recessive allele, heterozygous condition. So next, this is law of dominance. Next we have law of segregation. Law of segregation, what does mean segregation? Segregation means separation. So law of segregation describes how alleles segregate or separate during the gamete formation. In our introductory slide we have discussed that these alleles, this is gamete, this is cell, okay, and this will give rise to gamete. In this case, we have 2 alleles, capital T and small t. On gamete formation, what will happen? These 2 alleles will separate, okay, in each gamete. This will give rise to, this cell will give rise to 2 gametes. One is capital T and second will contain small t. The alleles are separated during gamete formation. 50% gametes will contain capital T and 50% gamete will contain this small t allele. This is what I have written here. It describes how alleles segregate or separate during gamete formation, and this process of gamete formation you must have read in reproduction chapter. Now each gamete receives only one allele for each trait from the parent. So in this case, this is not possible. This case is not possible that during gamete formation both the allele goes to only one gamete, and in second gamete there is no allele. This case never possible. During gamete formation, each gamete will receive only one type of allele. So we can say that there is 50% chances of having a capital T gamete and a small t gamete. This law of segregation is universally accepted. There is no exception in case of law of segregation. In law of dominance, we have certain exceptions which you will study in higher classes. But in law of segregation, we have no exceptions. This law is universally accepted. And this is very simple. This is all about law of segregation. Law of independent assortment. In law of independent assortment, it states that gene for different traits segregate independently during gamete formation. Okay, From law of separation, we know that during gamete formation, each allele is separated in the gamete. If we have another case that we have multiple character at a single time, let's say in this cell we have character for seed shape, we have this condition here. This is the character of seed shape. R stands for round and small r stands for wrinkled seed, and this is the character of plant height, and here T stands for tall and small t stands for short height, and this is the character of seed shape. During segregation, during gamete formation, what will happen? How will these two traits segregate? How will these alleles segregate during gamete formation? For single trait, we know that each allele will be transferred to a gamete, okay, to a different gamete. Alleles are separated. Now if we have another condition in which we have more than one trait, in this case we have allele of seed shape and allele of height. So how will they separate? They will be separated same like this. Let's say we have two gametes. Two gametes are formed from this cell. In one cell we will be having R. In second cell we will be having small r. We have separated one trait in gametes. Now it's a turn of second trait. How will this second trait separates now? Okay, in same way. One will enter into this gamete, second one, small t, will be transferred to this gamete. So now if you can see, there is no difference in the separation of alleles in the gamete formation. Okay, when we had only one trait, what we have seen that one allele will goes to one gamete and second allele will goes to second gamete. When we have more than one trait, similar thing is followed. Here capital R, one allele of seed shape will go into one gamete, and small r, the second allele of seed shape, will enter into another gamete, and capital T will enter into one gamete and small t will be transferred to this another gamete. So segregation, the separation is same. So here we have one thing, that this trait of height doesn't influence the transfer of another trait. Another trait is of seed shape. So the trait of height doesn't influence the transfer of seed shape character during gamete formation. It doesn't matter how the this character is transferred, how the plant height character is transferred into gametes, it doesn't matter for seed shape. Okay, these two events are independent of each other, means this can be transferred in this way also, like this, like this. Okay, both cases are possible. These two separations are independent of each other. These two traits will be transferred without any influence of each other. So if you see example here, here we have parent. In parent we have RR, capital RR, small YY. Wait, RR represents round, YY represents green. This is the color. And here we have shape. Shape is wrinkled and yellow. RR round is, and this is the wrinkled and yellow. If you can see here that yellow color is dominant over green and round shape is dominant over wrinkled. So during the gamete formation, this R is transferred to this gamete, and this Y is transferred to this gamete, and this R is transferred to this gamete, and Y is transferred to this gamete. These are the gametes, and this is the progeny, this is the offspring we have on crossing, and you have to write this way only. You can't write this thing like this. You have to write alleles of a trait together. So here we write round. So we have this RR, capital R, small r, capital Y, small y. So what is the phenotype of this case? We will be having round and yellow phenotype, which means that this round is dominant over small r, that is wrinkled, and this yellow is dominant over green. So this is how the law of independent assortment explains the transfer of genes for different traits segregate independently during gamete formation. Now, if we see independent inheritance, two separate traits, shape and color of seed. How can we study this? We can study this with the help of Punnett square. Now, what is this Punnett square? Punnett square is the representation of genotype. This RRYY, this genotype of allele, is the representation of genotype of offspring. Okay, progeny of a cross. Now here we have this cross, parental, This we have parental generation, okay, round and green, wrinkled and yellow. This is the phenotype, and this we have given here genotype, okay. So here we have capital R small y, capital R small y. From this, these are the gametes of these parents. Now these gametes will combine and will give rise to F1. That F1 is capital R small r, capital Y small y, which means round and yellow. Okay. Now further on self pollination. This F1 progeny is self pollinated. Means this is self pollinated. A cross is performed like this: R R Y Y R R capital Y, self pollinated. Now on self pollination of this F1 progeny, what will we get? We will get F2 generation, and in F2 generation we have these two, one this horizontal line, one we have this vertical line and this horizontal line, okay. And in these lines we have written here the gametes formed by these parents, okay. Let's say this is parent 1, this is 2. Here we have written gamete of 1 and here we have written gamete of 2. Now if we will cross these gamete, we will multiply these gamete cross. So what will we get from this? R capital R small capital Y, okay. Now in second, what will we get? Capital R, okay. Here we have capital Y, here we have small y. So here is the progeny will contain capital R, capital Y, small y. So we have to cross like this. These are the gametes, and on cross, what will be there? We have this genotype of progeny. So overall, this is the ratio what we finally got from this cross, from this dihybrid cross. As here we are considering two characters at a time during cross, so this is a dihybrid cross. What ratio we got? We got this ratio 9 is to 3 is to 3 is to 1. And in this ratio we have round yellow 9, round green 3, wrinkled yellow 3, and 1 is wrinkled green. How this ratio is attained? How does this explain independent inheritance? Let's say if we cross this R and small r, what will we get? We will get in first generation, we will get this thing, and on self pollination of this self pollination, what will we get? F1 generation. We will get F2 generation. In F2 generation, we will be having R, capital R, capital R, small r, small r, small r. This ratio of 3 is to 1. This is the ratio for this first trait, okay, for this trait. Now we have second trait, seed shape. Second trait is color, and this was the trait of seed shape. Now in the color we have yellow and green. Yellow is dominant over green. So same, we will perform monohybrid cross in case of color yellow into Y. In second generation we will get again this ratio 3 is to 1. This is the F2 generation ratio 3 is to 1. We have 3 is to 1 ratio for character 1 and 3 is to 1 ratio for character for trait number 2. How will we obtain ratio of dihybrid cross? These were the ratios of monohybrid cross, as we considered only one trait at a time. Here we considered C shape, here we considered color. Now if we see ratio of dihybrid cross means we are going to take both trait at the same time. What you can do here, you can simply multiply this monohybrid cross ratio. If you multiply here, what will you get? 3 3 9, okay. 3 into 1 3, then 3 into 1 into 3 3, and 1 into 1 1. And this is the ratio of dihybrid cross, what we have seen in previous slide here. This is the ratio what we are looking for. If on this cross where we have two traits and we are getting this ratio of 9 is to 3 is to 1 ratio, that means that those two traits are inherited independently. Means one character, one trait won't influence the inheritance of another character, another trait, okay. They are inherited independently. So this is the case, and this is how the dihybrid ratio comes on independent inheritance of two traits. Now you know law of segregation, law of independent assortment, law of dominance. Now here, how do these traits get expressed? How the traits are inherited? How the traits are inherited? Question here is how these traits are expressed now. Once the traits are inherited, next is expression. What is expression? Expression is function. Okay. How these traits are expressed in the body. Progeny has received this gene from the parent. Let's say this is the gene of plant height. Okay. Now, if it contains T, dominant allele. In each cell, there will be two copy of a gene. This is allele 1, this is allele 2, and this is the cell. And in cell, we have two alleles of a gene. So here, allele 1, allele 2. Now, these alleles will make protein in the cell. And this protein will act as an enzyme, and further this enzyme will regulate chemical reaction inside the cell and lead to the expression or function of the gene. This is how the traits get expressed. Enzyme here is responsible for releasing some chemical like phytohormone in case of plants. And then this phytohormone will help plant in growth, and that growth will increase plant height. So the level of plant height will tell us the expression of gene. Which form of gene is there in plant? Okay, if plant is tall, that means plant contains gene of tallness. And if plant height is short, what will we assume? That plant contains allele of short height. So this is how traits are expressed. This DNA, this alleles will encode a protein. A protein is made from DNA, and then this protein can function as an enzyme, and further this enzyme regulates various chemical reactions. For example, phytohormone formation, phytohormone production, and these phytohormone will next perform their function of expression. Okay, they will lead to different functions, like growth hormone will increase height of plant. So this is the case. How do traits get expressed? This is all. We have completed this chapter of heredity. In this chapter we have discussed about basic terms of heredity. What is gene, what is chromosome, phenotype, generation, F1 generation, P1 generation, all these things. And we have considered Mendel's work, monohybrid cross and dihybrid cross. Then we have studied different laws of inheritance, law of segregation, law of dominance, law of independent assortment. Okay, we have observed Punnett square, and you have to remember the ratio. You have to remember the ratios of cross. Okay, for monohybrid cross, in case of monohybrid cross monohybrid cross we have ratio of 3:1. Okay, this is phenotypic ratio. And in case of dihybrid cross we have a ratio of 9:3:3:1. You have to remember these ratios whenever you write monohybrid cross, dihybrid cross. You have to write these ratios as well. Okay, and you have to give examples of these crosses, and also you have to draw these crosses of monohybrid cross, dihybrid cross, considering any trait. Okay, you can consider flower color, you can consider plant height, tall or short character for drawing these monohybrid cross and dihybrid cross. Okay, understood? Now this was all about this Mendel's work. Now in next video part 2 of heredity, we will consider sex determination. Okay, so thank you, all the best.